Hello and welcome to this Qlucore webinar about Olink data and how you can import, pre-process and analyze Olink data in Qlucore Omics Explorer. My name is Jan Nilsson, I'll be your host today. There are many different Olink panels and you also got Olink software that can be used to export data that has been collected from the panels. And you can export normalized protein expression values, NPX values using the Olink software. And then you can import CSV files that you have exported into Qlucore Omics Explorer for exploration and analysis. There are different types of software. One is called Olink NPX Map, and that can be used to handle Olink reveal panel data. And you can export that data into CSV files in a vector or column-based format. We'll have a look at that in an example. You can also export NPX data from Olink High-Plex panels like Explorer, HT and 3K, and they can then be exported into Parquet files. They must be converted to CSV files before you can import them into Qlucore Omics Explorer. There is also software called Olink NPX Manager that can then handle target 48 and target 96 Olink panels and export data in CSV files, normally in a matrix format, but can also be in a vector format. And we'll have a look at a matrix format synthetic file and a vector file from NPX Map that is also going to be used for analysis. If we first look at Olink reveal data, here is an example where we exported it to a CSV file. The data is organized in a vector or column-based format. To the left you can see the sample identifiers in one column. And then here is also an annotation column called sample type where we can keep track of our control samples, for instance. Then we've got one column called Olink ID, which is then the variable identifiers, and we also got corresponding variable annotations like the UniProt identifiers. To the right you can see one column called NPX then, that's the normalized protein expression values. And we are going to import this in a synthetic file and see how we can work with it. One thing that's also important is that I mentioned that there are control samples. Those can be removed in the Qlucore product before you do the analysis. And we can also change between identifiers, and we can also remove those variable identifiers that are control variables in a dataset. We'll have a look at that. In this case we have a small file with those control variables AMP1, EXT1 and INC1 that we're going to remove using the set operation tool in Qlucore. I will now leave the presentation and we will go into the product instead and have a look at an example. I go to File, select Open with Wizard, and then I select in this case first the reveal example here. Data wizard starts. Type of data, it's other data. Count is if you have RNA sequence data. This is other data. Press next. Separator has already been detected, but you can change if there is some other. Press next. Data is organized as columns or vectors here. So we select vector, the ready button for that. Press next. Sample identifier, it's in this case this column. I just click on the top there and then I would get the name plus the content as well. Press next. Sample annotations, just using this sample type here, but there could be other columns that could have associations to the samples, then I can bring them in as well. Press next. Variable identifier, Olink ID. Press next. And also variable annotations, UniProt here, but it could be other things also that I would like to import and I can take them as variable annotations as well. Press next. And then data, I'll go to the right here, and there is a column called NPX, that's the normalized expression values. Press finish, data is imported, and we can after a while see a PCA plot here. We also have got here in the samples tab both the sample IDs and the sample types, and here you can see that we have the control samples as well. I'll start by removing them with the checkbox here, uncheck negative control, and you can see that the PCA plots opens up. I remove plate control as well and sample control, and then we just have the samples left here, and now we can start the analysis of them. But first, there could also be variable controls. Let's go to that. Variables here, got them here. I will now change in the tab data here from Olink ID to UniProt, and we will see the UniProt identifiers here. As I mentioned, in this case we have 3 control variables, and I will import them as a small file. You can of course search here as well and build a small list that you can use to remove them from the dataset. But I will import them. I've got a small text file where I've got those 3 variable control in the file, 1 each on a row. I select this icon here that looks like an arrow going into a tray here, and that I can use to import this text file with control variables. Press open here. Got them here, AMP1, Ext1 and Inc1. And now I will use the set operation here and do a difference between the dataset and the control variables and create a new list. And now I will apply that list directly here by the checkbox here. Close this also. So now we're just using 1034 variables, not the control variables. And now we are ready to go. So we have both removed sample controls and variable controls. So now we can set up statistical tests or explore the dataset. I will now leave this dataset here. I will close here. Discard the information. I will also remove the variable lists here. And now we'll talk about another type of data in Olink. Go back to the presentation here. And we'll talk about target data. For instance, target 96 and target 48 panels. And here we'll have a look at the target 96 synthetical data set here. It's then data that you normally export from Olink NPX Manager software to a CSV file, and in this case organized as a matrix. Here below you can see you have the information about the variables here on top. There are many more, in this case 96, but 92 if you have removed certain control ones. And then we got the NPX values here and the samples here. In this data set we got 30 samples that we're going to have a look at. We're also going to add an external set of annotations, in this case one annotation with 2 groups, and we'll see how we can set up a statistical test here using that annotation. Because in many cases you may have additional information in files that you can then add to your dataset as an annotation using the wizard for sample annotations. And we'll have a look at how you can work with that. But first we're going to import this synthetic file. So I leave now the presentation, go into the program again here, and we go to File and open with Wizard again. And now we are going to have a look at T96 matrix. Open the Wizard here. It's other data again here. Press Next. Separator has been selected. Now it's matrix data, not vector data. Press Matrix. Select Next. Treat rows as, well, it's samples here, so we change to samples. Data matrix starts here. Just click in that cell, then use Next. And then we have to go to the bottom right corner. Now I go to the right first, to the last variable here. You can see that we have some information to the right also that we're not going to import. Go down, we can see that we have some information at the bottom here, lot values and so forth. We're not going to import that. We'll stop here. That's the last NPX value. Press next. And then we'll bring in the sample annotations here to the left. And then we'll bring in the variable annotations that we have here on top. Press finish. Data is imported here, and we are going to actually work in another plot here. I've selected a heat map for this exercise, so we'll use the heat map instead. Now, in this case here, we will also change here in the data tab using SA as the identifier here. We've got the names here. And we will also, for this data set, make some other changes here in the heat map. So we will here in the View tab select here to order our variables here with hierarchical clustering here. And we will also do some changes to the ordering of the samples. But first, I will import the small file that I talked about with new annotation. I go to File, select Import, and then Sample Annotations via Wizard. And then I have a small file, a text file here with just 2 groups. Press Next here. We can see that we have one which identifies the sample and one which identifies the group that I'm importing. Orientation is rows here. Select Identifier here, same identifier as in the dataset. Press Next. And then I'll bring in also the annotation. Press Finish, and we'll have that annotation here as groups, Group A and Group B. Now I can order according to that also for the samples here. Instead of sample annotation 1, I select groups, so we order by that, and we can also select here to color by that annotation. We'll set that on top like this. Here I would like to have labels also using essays, so I will have added that as well. Let's say that I'm interested in the best discriminating variables in between Group A and Group B, then I can start the statistical test here. Select standard statistics. I can select the 2-group test. There are many of those to select in between. If you have normal distributed data, you can select a t-test as an alternative, Mann-Whitney if not. T-test. I use the annotation that I've imported called groups then. I would like to test Group B against Group A. I can use the slider for this. Or I can set a value, like a Q value of 0.05 as the threshold value. And then we can have a look in the variable tab here and then add information here for the variables here, like Q values. And I can order them from the lowest to the highest here. And I can also select to have the log fold change values here as well. Now, if I would like to export this, I can use this arrow going out from a tray, and then I can export my data. That's all I'm going to show. We have been looking at now target 96 data and how we can import that. That was a matrix-based format in a CSV file. And we, before that, look at Olink reveal data that has been exported using NPX Map into a CSV file. You're most welcome to come to our other webinars as well. And if you've got questions about Olink and so on, you can contact us at Qlucore. There is a form that you can use at our website. Thank you for attending the webinar today and hope to see you soon.